Hello and welcome. Today we are diving into one of the most incredible journeys in the entire human body, the visual pathway. We're going to follow a single particle of light from the outside world all the way until it becomes a fully formed conscious image inside your mind. It's a truly amazing trip. But have you ever really stopped to think about that? How does a simple flicker of light actually transform into something as complex and meaningful as a familiar face? Well, that's the big question we're going to break down in this explainer. Okay, let's get into it. So here's our roadmap. We're going to follow this visual signal through 5 key stages. First, the initial spark, then a stop at a critical relay station, a trip through a crucial crossroads, its arrival at the final destination, and then finally, how the brain actually splits the signal to make sense of it all. All right, so our journey kicks off right at the back of the eye, in this amazing light-sensitive layer of cells that you know as the retina. This is where the magic first happens. This is where light from the outside world gets translated into the language of the brain. It's this really precise 4-step chain reaction. So first, a photon of light slams into the photoreceptors. Yep, the famous rods and cones. They convert that light into a tiny electrical blip. That signal then gets handed off to the bipolar cells, which in turn pass it to the retinal ganglion cells. And it's the long tails of these ganglion cells, they're axons, that all bundle together to form the optic nerve. Basically, the superhighway carrying this new information out of the eye. Okay, so our signal is officially on the road, speeding down the optic nerve. Its first major stop is a structure deep inside the brain's thalamus called the lateral geniculate nucleus, or just LGN for short. You can think of this as the central sorting facility for vision. Honestly, the best way to think about the LGN is as the brain's super-organized central post office for everything related to vision. It doesn't create the final image, but it gets all the incoming visual mail, sorts it with incredible precision, and makes sure every piece gets routed to the right place for final processing. And what's so cool is that the LGN isn't just one big room. It's actually organized into 6 distinct layers. Layers 1 and 2, the magnocellular pathway, they're kind of like the bulk mail department. They handle all the big picture stuff, motion, and broad shapes. Meanwhile, layers 3 through 6, the parvocellular pathway, that's like the first-class mail department, handling all the information about color and fine, intricate details. And this post office is just ridiculously organized. First off, it only handles mail from the opposite visual field. So the right LGN is processing what you see on your left. It also maintains a perfect retinotopic map, which means that points next to each other on the retina stay next to each other in the LGN. And maybe most importantly, it dedicates a huge amount of its space to the macula, our crystal clear high-definition central vision. Now, before those signals from the retina can even reach the LGN sorting facility, they have to navigate the single most famous intersection in the entire visual system, the optic chiasm. This is where some serious rerouting is about to happen. Yeah, this intersection is the site of what's called the Great Crossover. Some of the nerve fibers are about to cross over to the opposite side of the brain, while others are going to keep going straight ahead. So, if you remember one thing about this, remember this simple rule. The nasal fibers, the ones on the side of your retina closer to your nose, they cross over. But the temporal fibers, the ones closer to your temples, they stay put. It's this crossover that allows your brain to combine the slightly different views from both of your eyes into one seamless 3D image. And this anatomy perfectly explains why the chiasm is so important in medicine. If something damages just those crossing nasal fibers right at that intersection, you get a very specific kind of vision loss called bitemporal hemianopia. Basically, you lose your peripheral vision in both eyes. It creates a tunnel vision effect. And why would that specific spot get damaged? Well, it all comes down to location, location, location. The optic chiasm sits in some prime, but very crowded real estate. It's located directly above the pituitary gland. So if a tumor starts to grow on the pituitary, it can press straight up and squish only those crossing nasal fibers, leading directly to that classic tunnel vision. And here's a quick little myth-busting moment for you. For decades, anatomy books taught about a little loop of nerve fibers here called Wilbrand's knee. But it turns out modern research suggests it doesn't actually exist in living people. It's likely just an artifact, a distortion created when preserving brains for study. It's a pretty fascinating example of how science is always updating itself. Okay, our signals have successfully made it through the crossroads. Now they travel in bundles called the optic radiations, on the final leg of their journey. to the primary visual cortex, located all the way at the back of the brain. This is where the raw data finally starts getting decoded into what we call sight.
But the fibers don't all take the same route. The inferior fibers, the ones carrying information from the upper part of your vision, they actually take a little scenic detour. They loop forward through the brain's temporal lobe in a path called Meyer's loop. And because we know this anatomy so precisely, we know that if there's damage specifically to Meyer's loop, it results in the loss of the upper outer quadrant of your vision. Clinicians have a great nickname for this, a pie-in-the-sky defect. Meanwhile, the other set of fibers, the superior fibers carrying info from the lower part of your vision, they take a much more direct route, straight back through the parietal lobe.
And you can probably guess what happens here. If there's damage along this more direct path, it causes the opposite problem, loss of the lower outer quadrant of vision. And yep, this one's called a pie-on-the-floor defect. It's an incredible example of just how precisely our brains are mapped out. So our signal has finally arrived, but believe it or not, the job still isn't done. The second that information hits the primary visual cortex, the brain immediately splits it again, sending it down two different processing streams to figure out what exactly we're looking at. These are the famous what and where pathways. The where pathway sends info up to the parietal lobe to process things like motion and spatial awareness. Where an object is and if it's moving. At the exact same time, the what pathway sends that same info over to the temporal lobe to process things like facial recognition, color, and fine detail, what the object is. So, what an incredible journey, right? We followed a signal from its birth in the retina, watched it cross over at the chiasm, get sorted at the LGN post office, then tracked it all the way to the visual cortex where it was finally decoded by the what and where streams, giving us this rich, seamless visual world we live in. And all of this brings us to a final, pretty mind-bending thought. This whole complex process makes one thing really clear. You don't really see with your eyes. They're just the cameras. You see with your brain. So that begs the question, what are you actually seeing? Are you seeing objective reality, or are you just experiencing the incredibly detailed story your brain constructs for you? Something to think about. Thanks so much for joining me.